Thank you. So to explain exactly what it is I've done, I think I need to start with some background information. First of all, I'd like to talk about a method of genetic engineering known as CRISPR-Cas9. CRISPR-Cas9 was developed in 2013, so it's a relatively new method, but it has been found to be so far the most effective and now widely used method for genetic engineering. CRISPR-Cas9 has two main elements, the guide RNA and the Cas9 molecule itself. The guide RNA, as the name suggests, guides the molecule to a location in the genome where the Cas9 molecule can then edit the genome. It can modify, delete, or insert bases so that it can change the genome in many different ways. In 2015, a research at, I believe, MIT found that there are two loops protruding from the Cas9 molecule, seen here in red. And these loops, because they protrude out from the molecule, you can add an additional loop to these and then add molecules which can affect how the Cas9 molecule functions. Another important concept for my research is the epigenome and epigenetic modification. The epigenome, most people have heard of it, most people don't understand what it is. It's basically what controls gene expression rather than the genes themselves. An important aspect of the epigenome are histones, which there's a visualization of here. Histones are protein molecules that DNA is wound around. The primary functions of histones are not only to compress DNA so that it fits into the nucleus, but also to control gene expression, which is more relevant to my topic. Two ways in which histones control gene expression are acetylation and methylation, which are, as the name suggests, addition of acetyl and methyl groups to histone tails. In my project, I work quite a bit with two molecules that control these processes, EZ, which controls methylation, and CBP, which controls acetylation. In general, it's been found that acetylation increases gene expression, whereas methylation has the opposite effect, so it decreases gene expression. The organism that I am genetically modifying, in a way, is Drosophila melanogaster, which is the common fruit fly. Specifically, I am working with S2 cells, which are derived from embryos of fruit flies that are about 20 to 24 hours old. The genes that I'm targeting are twist and SOG genes, so I have two separate experiments, one with the CBP molecule and one with the EZ molecule, CBP targeting the twist gene and EZ targeting the SOG gene. So now that I've explained a bit about the concepts behind my project, what is it that I've done? Basically, I've tested a new method of gene regulation. So rather than modifying the genome as CRISPR-Cas9 traditionally does, I've attempted to create a new molecule that can regulate gene expression with epigenetic modifiers, which are CBP, Krebs binding protein, and EZ enhancer of zest, by fusing these molecules to Cas9. The first step in this method was to create plasmids that contain the proper DNA sequence. So the DNA sequence of a Cas9 fused to a guide RNA, an MS2 loop, and a CBP or EZ molecule. Plasmids being a word for small DNA molecules. So in order to do this, I amplified the DNA sequence, inserted it into plasmids, and then inserted those plasmids into E. coli bacterial cells. After allowing the cells to grow for about 48 hours, I used 3 different methods to check the quality of the DNA, or quality, concentration, and if the plasmids have the correct sequence. One way in which I did that is a picture up here. It's called an agarose gel electrophoresis. There's a DNA ladder here, and the bacterial colonies that have the correct plasmid sequence are lit up here, 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 and here. In here. So basically those were the colonies that had the correct sequence and could be further used in the next step of the method, which was the actual modification of the epigenome. So I extracted the plasmids from the E. coli cells and then put them into S2 cells, so fruit fly embryos. I allowed those cells to grow for 48 hours and then extracted RNA from them. The RNA was converted to DNA, complementary DNA, and then the final step of the method was qPCR, which stands for qualitative PCR, which calculates gene expression levels. The results I received, you can see here, the results are grouped into 3 different sections by what section of the SOG gene were edited. It's useful to note that these are the results for the EZ experiment and that the results for the CBP experiment will not be in until later today, so I couldn't show them here. The results are grouped, as I said, into 3 sections based on what section of the SOG gene were targeted. I targeted the SOG promoter 2, SOG promoter 3, and SOG enhancer 3. What these sections do isn't so important to the experiment, but the reason why they are targeted is that prior research has found that epigenetic modification in these sections of genes usually seems to work well. The first bar in each group, so the beige-ish one, is a control, so those conditions don't contain any plasmids and are ordinary levels of SOG gene expression in fruit flies. The second bar in each group is the plasmid containing a Cas9, guide RNA, MS2 loop, and EZ molecule, so that is the experimental condition. And the third and fourth in each grouping are various controls that contain incomplete Cas9 EZ complexes, so some of the components but not all of them. So to interpret these results, unfortunately it is quite clear that at some point during the experimental procedure the plasmids were cross-contaminated with each other, so the results aren't of much use. The way I can see this is, for one, various controls appear to have an upregulating effect or downregulating effect, so that they are higher or lower than the control, which shouldn't really occur because the complex that is modifying the epigenome is not complete. So, for example, this plasmid contains an MS2 loop and a guide RNA, and a Cas9 molecule, but no EZ molecule. The other way I can tell that there was cross-contamination during the experimental procedure is that here, EZ, this is the complete plasmid, appears to have an upregulating effect, so gene expression is increased compared to the control. As I mentioned earlier, since EZ is involved in methylation of histones, it is highly unlikely that it will have an upregulating effect, but it is more likely it should have a downregulating or possibly no effect. So, unfortunately, I came to the conclusion that this experiment needs to be redone to have proper results, but hopefully the CBP experiment that I have completed, that is done today, should have better results. Yeah, I guess that's it. I'd like to thank my mentors, Dr. Suresh Sajwan, Professor Mattias Mannevik, Ray's partners, Kjell och Märta Beyers Stiftelse, AstraZeneca, and of course, Race for Excellence.